ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3-carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
Cells break down food molecules, such as glucose, through multi-step pathways. For example, in the process of glycolysis, breakdown of glucose molecules releases energy that is captured by the energy carrier molecules ATP and NADH. A breakdown intermediate, pyruvate, is imported into mitochondria, where it is converted into acetyl-CoA and fed into the citric acid cycle. Acetyl-CoA can also be generated by breakdown of fats or amino acids. In this circular reaction path of the citric acid cycle, carbon atoms are burned, that is, oxidized, and released one by one as the waste product carbon dioxide. In this way, energy is released stepwise and captured by energy carriers, including NADH. NADH funnels energy to the electron transport chain in the inner mitochondrial membrane. This fuels the proton gradient that is then used for the production of ATP, the cell's primary energy currency. The molecule that enters the citric acid cycle is the 2-carbon compound acetyl-CoA. Acetyl-CoA joins with the 4-carbon oxaloacetate to create the 6-carbon citrate. We'll track the carbons from acetyl-CoA with a red color. The 2 carbon atoms from oxaloacetate marked in blue will be released during this cycle to form carbon dioxide. In the next step of the cycle, citrate rearranges to form isocitrate. Note that the hydroxyl group is in a different position in these two molecules. In the next step, energy is captured by an NADH molecule, and a molecule of carbon dioxide is released. In this reaction, isocitrate is converted to alpha-ketoglutarate. The hydroxyl-bound carbon is stripped of its hydrogen atoms, resulting in a carbonyl group. One of these hydrogen atoms is picked up by NAD+ to form NADH, and another is released as a proton. The carbon and 2 oxygen atoms are then released as CO2, creating the 5-carbon alpha-ketoglutarate. The next reaction also produces NADH and releases CO2. The alpha-ketoglutarate from the previous reaction is converted to succinyl-CoA by the addition of coenzyme A. The enzyme for this reaction adds a high-energy thioester bond to coenzyme A, releasing the carbon and 2 oxygen atoms and converting NAD+ to NADH. The next reaction releases enough energy to form GTP, an energy-carrying molecule related to ATP. In this reaction, succinyl-CoA is converted into succinate. The release of the coenzyme A group provides the energy to combine GDP and inorganic phosphate into GTP. Note that succinate is a symmetrical molecule. The 2 end carbons are chemically identical, and the 2 carbons in the middle are chemically identical. For convenience, we will continue tracing only the 2 carbons depicted in the upper half of the molecule. In the next step, a molecule of FADH2 is produced. FADH2, like NADH, is an energy carrier that feeds high-energy electrons to the electron transport chain. In this reaction, succinate is converted to fumarate. Hydrogen atoms from succinate are stripped off and donated to FAD to produce FADH2. In the next reaction, fumarate combines with the water molecule. The resulting molecule is malate, with the water molecule added across the 2 central carbons. The next step produces the final NADH molecule. In this reaction, malate is converted to oxaloacetate. The carbon carrying the hydroxyl group is converted to a carbonyl group. This reaction releases hydrogen atoms and converts NAD+ to NADH, releasing a proton and producing the 4-carbon oxaloacetate. Oxaloacetate is thus replenished and can take part in another cycle, returning to step 1. Note the new position of the red carbon atoms, which originated from the acetyl-CoA in the previous cycle. In subsequent cycles, these carbons will eventually be lost as CO2. The green labels indicate the positions of the new carbons added during this new cycle.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Presenting the pentose phosphate pathway.
We have many very cool images to show you, starting with the one in the background. This is our starting point for the pathway, and as we bring everything up into full screen, we'll get started talking about the very exciting pentose phosphate pathway. So this first molecule is glucose 6-phosphate. It's important in the first step of the oxidative phase of this reaction. The oxidative phase is very linear, so we can go through this kind of quickly and hit a good review for it. So glucose 6-phosphate is transformed into 6-phosphoglucoacetone. This is catalyzed by glucose 6-phosphate dehydrogenase, which is shown on screen right now. You can see this is a fairly large enzyme, and the binding pocket is right there in the middle in sort of the X-shaped region formed by a bunch of beta helices, or beta sheets, excuse me, not beta helices. That would be kind of groundbreaking. In the middle of the beta sheet region is the binding site, as we've already covered. Now, this is a dehydrogenation reaction. The hemiacetyl hydroxyl group on carbon 1 of glucose 6-phosphate is turned into a carbonyl group, which generates a lactone ring in 6-phosphoglucoacetone, as shown. There's our lactone ring. NADPH is generated in this reaction, and that's the most important point, is that's an important reducing reagent in many cellular cycles. Now, this takes place in the next reaction in the cycle, the second one overall, where it's transformed into 6-phosphogluconate. This is catalyzed by 6-phosphoglucolactonase. And it's a very simple hydrolysis reaction, but does need a fairly hefty dimeric enzyme in order to carry it out. In a hydrolysis reaction, H2O, water, is used, and simply protons are generated in that reaction. Our 6-phosphogluconate is changed into ribulose 5-phosphate in the last reaction of the oxidative phase. This is catalyzed by 6-phosphogluconate dehydrogenase. This is 6-phosphogluconate shown on screen right now, which is the first reagent in the step. The next step is catalyzed by 6-phosphogluconate dehydrogenase, which is one that we couldn't quite find. But it's an oxidative decarboxylation reaction, where NADP+ is used as an electron acceptor. And this step of the reaction generates NADPH, CO2, and ribulose 5-phosphate. This is the first step in the pentose phosphate pathway in which the carbon balance is changed. And that's just shown through the CO2 given off. I'm going to throw it over to Xander, who's going to go over sort of the overall reaction during this oxidative phase. First, we're going to get ribulose 5-phosphate up. As you see, this is a 5-carbon sugar in difference to the 6-carbon sugars that we've already got up.
Okay. Well, now as we can see from the carbon balance that is currently displayed on the screen here, this is the oxidative phase of the pentose phosphate pathway. Excuse me. And it is fairly similar to most oxidative phases of most metabolic processes, including glycolysis and the Entner-Dudoroff pathway. In short, a glucose derivative is reduced to a smaller carbon derivative, releasing CO2, and at the same time generating NADPH for use in metabolic biosynthetic pathways.
So now we'll enter into the more complicated point of this reaction as we get everything off the screen there. So now we're headed into the non-oxidative phase. This is more non-linear. There are a couple different branching points. We'll try to cover these as best as we can. But we start with our good friend here, ribulose 5-phosphate. This can be changed into one of two different products. The first product is ribose 5-phosphate. And this is done via ribulose 5-phosphate isomerase. And that's a pretty simple reaction. As you can see, it uses a fairly simple enzyme to do this. It changes the structural formula without changing any of the atoms in the molecule.
In particular, it changes L-ribose into D-ribose.
The other possibility for this reaction is that ribulose 5-phosphate can be turned into xylulose 5-phosphate via ribulose 5-phosphate epimerase. Now, if you haven't heard the term epimer before, it's just a shifting of hydrogens simply. If a hydrogen is in the top position in one of the epimers, it's in the bottom position in the other epimer. It's just a very simple change. And this brings to mind a popular saying that they don't think it be like it is, but it do.
By Oscar Gamble.
It's a very valuable bit of information to learn from that simple reaction. Now I'll enter even more complicated. This was just a simple one branch. Now we're going to have two different reactants enter and create two different products. Our first set of reactants is both of the steps of this reaction. Xylulose 5-phosphate and ribose 5-phosphate will combine in order to form glyceraldehyde 3-phosphate, which is, your eyes should light up, your mind should light up. You should know quite a bit about glyceraldehyde 3-phosphate. The other product it produces is cetoheptulose 7-phosphate. Now, glyceraldehyde 3-phosphate, a very important molecule, and this can be combined together. Two of them can be combined to form fructose 6-phosphate, which is used in glycolysis. And that's kind of where this feeds back into some of the other pathways. Now, this sort of branching reaction are two 5-carbon molecules combining to form a 3-carbon and a 7-carbon molecule. This is done via transketolase, is the name of the enzyme, and it does exactly what it sounds like it does. It'll transfer a keto group from one of the 5-carbon molecules into the 7-carbon molecule. This is the transketolase enzyme. As you can see, it's very large, and it includes many alpha helices. The next stage of this will take those two products, our cetoheptulose 7-phosphate and our glyceraldehyde 3-phosphate. So again, there's 10 carbons entering, and it will change them into erythrose 4-phosphate, which is an important precursor to several amino acids, but we'll get more to that later. And it will also form fructose 6-phosphate. And fructose 6-phosphate, as we said, is simply two glyceraldehyde 3-phosphates melded together. And that'll form fructose 6-phosphate, which goes into the glycolytic cycle. This is done via transaldolase, which is another fairly complicated enzyme. As you see, this one is even perhaps more complicated than the other one. It is a dimeric structure, as you can see. It's actually a fairly cool little, it's like a dimer of pentamers, if you will.
I almost see a smiley face right here. See? Smiley face.
I was actually thinking just individual flowers almost at the center.
Aside from that, it is a spectacular enzyme. Very complicated and very interesting.
And then the last reaction that we're going to go over is our only 9-carbon reaction. Xylulose 5-phosphate and erythrose 4-phosphate will form glyceraldehyde 3-phosphate and fructose 6-phosphate. This is basically just maximizing the amount of glycolytic products that we can get out of this cycle. This is done via the same transketolase enzyme, I believe. And that'll basically take us all the way through the pathway. All these branching enzymes, it's really important to note that in the reactions, the carbon balance is exactly the same. There's 2 reactions that are 10 carbons each, the first 2, and the last reaction is 9 carbons on each side of the reaction. So if you keep track of your carbons, the pathway becomes fairly linear and fairly easy to understand.
The pentose phosphate pathway is not so much a catabolic cycle. It is more of a anabolic cycle. Honestly, it creates products that are necessary for other metabolic cycles and and in particular some very important biosynthetic cycles. For instance, glyceraldehyde 3-phosphate, a very important molecule in the entire cycle, as shown here, feeds into amine synthesis and several other very important cycles. Some of the primary functions of the pentose phosphate pathway is it generates reducing equivalents such as NADPH, and it produces ribose 5-phosphate, which is used in synthesis of nucleosides and other nucleic acids. Furthermore, the production of erythrose 4-phosphate is very important, as it is a molecule that's used in the synthesis of aromatic amino acids. And furthermore, the pentose phosphate pathway, in fact, accounts for 60% of all NADPH formation in humans.
So this is a very important pathway in basically all living organisms that it occurs in. And it's been really great to be able to bring some of this to you.
We hope you've enjoyed our cast, and hope you'll tune in next week for our segment on DNA replication. Anyway, thank you very much, and have a good night.